Okay, is that working out? Okay, so today we have Liz Suter from Molloy University. Tell me if I'm not saying that right. I should have asked first. Awesome. And she'll be talking about oyster pathogen monitoring using third-generation sequencers. And I think I'm going to give a little bit of a lead up as to like why we were interested in supporting work on oysters and what's happening here in New York when it comes to oyster restoration in general. So this project was interesting to us for a couple different reasons. To us, there are different aspects. This sort of sits in the middle of a couple of different critical water resource issue themes that we have here at WRI. And I'll make the case that both climate resilience and aquatic ecosystems are themes that oysters belong within. And I'll do that in just a couple of slides. This just gives you a really quick snapshot of some work happening here in New York. So in terms of climate resilience, why are oysters and the restoration of oyster reefs important for climate resilience? On the right side of the screen here, I don't know, can you see my cursor? This is the Governor's Office of Storm Recovery, or GOSR, and they have the office within the Office of Resilient Homes and Communities. And what you can see here is something called part of the Living Breakwaters project. So this is actually, a rendering of a map off of Staten Island, where they're proposing to use this concept of reefs and oyster reefs and sort of a natural habitat to essentially dampen, things like wave energy and sort of erosive forces coming in during storms on those sort of shoreline communities. And so, you can see here down just a little snapshot of what they say. Oyster reefs are a natural storm barrier. So systems in New York Harbor were once a natural defense against storm damage, softening the blow of large waves and reducing flooding and preventing erosion. So you can imagine these structures out off the shoreline is essentially, dampening some of that wave energy that's coming in and heading toward the shore. And if you're interested in learning more about this, I would encourage you to check out the Billion Oyster Project, which Liz may talk about a little bit more. And so I just wanted to call them out as a group that's really doing some active work here in New York and has gotten attention, from the governor's office and from lots of other stakeholders, and is sort of a big deal in the New York, New Jersey harbor area. So that's the climate resilience part of it. Why is this important to ecosystems? This is maybe a little more obvious. This is a snapshot of the Hudson River Estuary Action Agenda. So again, this is a state document where they're trying to essentially look at the Hudson Estuary. So that goes from Albany in the north all the way down to New York City in the south, and trying to lay out some goals and strategies for sort of achieving ecosystem restoration and general estuary ecological and community health. And one of their strategies is to assess and prioritize management options and restoration opportunities generally. And within that, they talk about evaluating the use of restored oyster reefs as sort of having this conservation benefit, right? So oysters being part of the natural ecosystem in the harbor and estuary area, the idea of restoring those as part of this action plan, part of this state management plan. So maybe a little more specifically, right? Here's the New York, New Jersey Harbor and Estuary Program, which you can sort of think of as a more localized, a more focused action agenda. And they similarly call out oysters in their objectives, where they talk about improving the quality and likely success of habitat restoration, which again does apply to many species, but certainly applies to oysters. And I think my last slide in terms of kicking this off is really this one, and hopefully I'm not stealing your thunder here, Liz, but within that New York, New Jersey action agenda, they talk about oyster reef restoration goals, and they say that they're proceeding more slowly than other restoration goals, when they look at different things that are happening. So what you can see here, so this is a bar chart, and on the x-axis is essentially their assessment of whether they're reaching their restoration goals. So 100% doesn't necessarily mean that they've restored 100% of the habitat, but whatever their goals were between 2009, there's a little typo here. Oops, there we go. Whatever their goals were, they feel like they've achieved 100% of those goals when it comes to coastal and maritime forests, public access, and things like habitat for water birds. So according to their goals, they did pretty well. But you can see as you go up here, they're maybe not achieving some of the goals they hoped they would in sort of a 10-year time span. And wetlands, habitat for fish, crab, and lobsters, and oyster reefs are things that maybe haven't progressed as much as some of these groups would have liked. And so I'll just leave with a question there, which is like, we're interested in, why are oyster reefs and some of these other issues not progressing the way we might have liked or might have thought? What are the issues or barriers when it comes to oyster reef restoration? So there, I will stop and introduce Liz, or tell Liz to introduce herself.
Thank you. I'm going to share my screen now. Can you see that?
Looks good.
Okay, great. So that was a perfect introduction. Thanks, Brian. So let me jump right in. Well, actually, before I jump in to this project about monitoring oyster pathogens using a new generation of sequencers, let me acknowledge two of my students who I think are in the Zoom room right now, Matt and Joe, who have done the majority of the work for this. Especially the dirty work, getting out in the oyster reefs, collecting the samples, and also a lot of the wet lab work. So kudos to them. This is going towards their undergraduate thesis. Okay, so why do we care about restoring oysters? So Brian did a really great job highlighting the importance of oysters to things like climate change resilience and water quality. I wanted to show this quick video from YouTube while I talk about this to portray the ability of oysters to filter water. It's really amazing. So first, let me establish that oysters currently comprise less than 0.01% of what their historical numbers were a few centuries ago in New York Harbor specifically. And this is because of a lot of things. One is simply overfishing. They've been extracted at a really high rate and we're never able to come back. But now that we're trying to restore them, it also seems like they're coming back to an environment that wasn't their natural environment from a few centuries ago. So it's really hard for them to get reestablished in New York Harbor. There also are probably things like toxins and pathogens, which I'll get into. So we want to know as Brian said, what is the thing that's really preventing oyster restoration in New York Harbor or slowing it down? The reason why we want these guys to come back is one, because they filter water really, really fast. So in this video, it does like a 40-minute time lapse, and you can see how turbid the water is at the beginning and then gets cleared within 40 minutes just from this one little cluster of oysters. So a single oyster can filter several gallons of water per day. While they do that, they remove particulates and they deposit them in or near the reef, which ultimately clears the water, but that's also clearing a lot of other stuff that are on those particulates, like organics and nutrients and potentially toxins or pollution. So they're really great for water quality. There's also evidence that they're really great for nutrient removal, and then also really good for shoreline protection. So there's a lot of interest right now in building or helping oysters to build their reefs in places where we get a lot of wave energy, especially during storm surge, during a hurricane. And that area in particular that Brian highlighted on southern Staten Island was really impacted by Hurricane Sandy. And they believe that if those historic oyster reefs were there, as they were meant to be, the impacts from Hurricane Sandy would have been much less. That area in Staten Island also is historically was a big oyster fishing village. They played a big role socially there as well. So oyster restoration is the focus of a lot of nonprofit groups, including groups like the Billion Oyster Project, but they're having variable successes in different places. And what is driving whether an oyster reef is successful or not is really unclear. So part of it is probably environmental parameters. You know, oysters might be sensitive to temperature or salinity, which can vary in different locations. Oysters also require, in order for an oyster larvae to successfully sit on the bottom, it requires a particular substrate to sit on, and usually that's another oyster's shell. So a lot of the restoration organizations, you might have seen this, they collect oyster shells from restaurants and they put them out to try to increase the substrate for wild oysters to recruit on. So part of it is what substrate is available. Hydrodynamics are also important. So how the water flows around an oyster reef can determine things like how much oxygen is available. And oysters are animals just like us, they need oxygen. And so that might be limited in certain areas because of different environmental issues. And then another potential reason for this variable success is the microbiology. So oyster pathogens or causes of disease in oysters have been studied for a long time. And on the East Coast specifically, they've studied these two diseases called dermo and MSX. But these are eukaryotic pathogens. And there's a lot less known about prokaryotic pathogens. So prokaryotes, mainly I mean bacteria. There have been, Brian,
Hey, Liz, your slide may not be advancing. We're still on the 40-minute video.
Okay. Oops.
There you go. Thank you.
Thank you. So there's this increasing awareness that, well, all animals have a microbiome, right? And there's this increasing awareness that an individual animal's microbiome is really important for their health. And you hear about this with humans and their gut microbiome. And potentially in a microbiome, whether it's of an oyster, of a human, there's a potential for a microbiome to become what's called dysbiotic. So normally, under normal healthy conditions, we have a nice diverse microbiome. That includes opportunistic pathogens, which may not infect under normal conditions, but if something changes, those opportunistic pathogens that are in your microbiome could start to become pathogenic. So that's what they're showing here. These pathobionts are these things that hang around in the microbiome that have the potential to be pathogenic, and they can become abundant under certain conditions. Also, when a microbiome becomes dysbiotic, usually you just see reduced microbial diversity, and you can have a loss of the microorganisms that are beneficial for you. So we're aware of some species that are acute pathogens of bivalves and other animals in the marine environment. But it's less clear what sort of those opportunistic ones that might be driving dysbiosis. But in this project, we're sort of I'm interested in all of these, anything that could be driving a less healthy microbiome that ultimately might be the cause of some of the mixture of successful oyster restoration projects. So in the Hudson River estuary specifically, why might an oyster have a dysbiotic microbiome? Well, like I mentioned before, we're trying to reintroduce them into an environment that they've never really lived in before. The microbial ecology of the Hudson River is completely different than it was 300 years ago, and it's largely influenced, it's heavily influenced by humans, right? We have sewage runoff, we have just regular runoff. And so there's a lot of microorganisms there that didn't used to be there. So are these challenging the oysters in some way? And in particular, are they changing the microbial diversity in their microbiomes? So those are the broad questions we were asking in this project. We set out to really establish a method for looking at this. And so most of this project is methods development, but specifically, we were targeting 2 locations that are part of the Billion Oyster Project. So we partnered with them to get some oysters, and they have many locations around New York Harbor, as well as in Jamaica Bay and Long Island Sound, and they keep expanding beyond that. But these 2 locations, Lemon Creek Lagoon down south here and Sunset Park in Brooklyn, were of interest because This one in Sunset Park is right next to a sewage outfall, so there's potentially a lot of introduction and persistent introduction of human associated microorganisms. And Lemon Creek Lagoon, they've been monitoring the temperature for a long time, and they noticed that the temperature is kind of elevated because it's a shallow lagoon. And pathogens really like higher temperatures. So I wanted to look at these two specifically. Since the beginning of the project, we found that a PhD student at Rutgers, Heidi Ye, was also working with the Billion Oyster Project, interested in microbiomes for other reasons, but we've been sharing samples with her, and so she also gave us some samples from other locations, all within the same watershed and part of the Billion Oyster Project. Okay, so how do we usually detect species in a microbiome without getting too complicated? So normally, bacteria are distinguished phylogenetically by looking at the sequence of this particular gene called the ribosomal rRNA gene. And this gene is important for the functioning of a cell. If you don't have a good ribosome, the cell's not going to live. So a major mutation will never, if you have a major mutation, the cell won't survive. So it's a conserved gene, but it also has, so it has these blue regions, which are conserved regions, but it also has these very variable regions called variable or hypervariable regions. So what's great about this is that you can design genetic targets in the conserved regions. So these should look the same in all bacteria, and then amplify the space between those targets to look at the variable regions. And the variability is how we determine the difference between species. And so this approach has been used for several decades now to look at the difference between bacterial species and genera. And usually what we're looking at are small chunks of this gene. So each of these little arrows is like the location of a target or a primer, and they amplify what's between them. So usually they're looking at pieces that are between 150 and maybe at most 500 base pairs long in this area called V3, V4, V5, these three hypervariable regions. And so this is usually what's used in the most common type of sequencing, which is second generation sequencing. And what's great about this is we can sequence millions of chunks of these short chunks, they're called short reads, and get to look at the microbiome from sort of a meta view. The con is that these are only short reads. And so this is a major challenge for looking at pathogens, because when you only have a short read, you might only be able to distinguish two different genera, two different or individuals in a different genus, rather than different species. So we can't get to the species level necessarily. And with pathogens, it's also important to sometimes get to the level of strain. So after species, you can also talk about different strains. And you can't get that with these short reads. So the approach that we use in this project is a third generation of sequencers, which I've called 3G sequencers. And specifically, we use this Oxford nanopore sequencer called the MinION. So what's great about 3G sequencing, well, there's multiple things that are great about it. First, it can generate those long reads. So the reads can go up to 5,000 base pairs or even longer. And so in this project, we use an approach from Lee Kirchhoff at Rutgers that covers the entire gene, which is the 16S portion of the ribosomal gene, plus another chunk of the ribosomal gene called 23S. So it's going from here, where this says forward primer, to reverse primer. So this is 4,200 base pairs long, compared to in the previous slide, we were averaging around 200 base pairs long. So this gives you great taxonomic resolution, which you need in order to detect pathogens. The other really cool thing about 3G sequencers is they're cheap, and they're small, and they're really modular. So for 2G second generation, we used to extract our DNA and send our samples to a sequencing facility and get data back within a few weeks. But now these are small and cheap enough that we can buy them and use them in the lab ourselves, and potentially restoration organizations can do this too. And so this whole project has been designed with restoration organizations in mind in order to make this something that is hopefully one day doable by practitioners of restoration, not just in science labs. So so we were able to purchase one of these and get it working to sequence those long reads and give us our high taxonomic resolution. Another thing about these long reads that's really great is one of the hesitations about moving to the nanopore has been that it has a higher sequence error rate. So it gets about a small percentage of the base pairs wrong. Whereas 2G has a much lower error rate. But I'll say two things about that. One is the error rate in the last 2 or 3 years has come way down to less than 1%. And the second thing is that by sequencing the entire 4200 base pair gene, you overcome what you lose you overcome that loss in that because of that error rate. So we really can get that taxonomic resolution. Okay, so the first major part of this, and what Matt spent a lot of his summer doing, was optimizing a DNA extraction method. And this was challenging because we wanted a method that was easy, again, thinking about restoration practitioners using this in the future, but also we needed to find a very gentle method because we needed to be able to extract long pieces of DNA, whereas most DNA extraction methods are kind of rough, like they require you taking tissue and cells and putting them in a tube with these like glass beads and shaking them on a vortex. We couldn't do that because we didn't want to shred our DNA because we needed those long pieces. So he went through many different protocols trying to find the easiest protocol that gave us the most consistent DNA extraction results. So first he tried just lysing the cells by heating them. That was not great. He added a cleanup step after that. Also, and we still didn't get great results. Something called polyethylene glycol, which is a relatively easy method, but also wasn't super consistent. Then we moved to DNA extraction kits, which have these little spin columns to purify DNA. The first one was called DNeasy. The second one we used was specific for bivalves, called EasyNA mollusks. So they both use similar approaches. But again, they were not giving us consistent DNA extract. They would work sometimes, but not for every individual oyster. Then finally, we did a digestion with enzymes, and that was okay. But then when you added that with the phenylchloroform extraction step, we were consistently getting DNA from these oysters of sufficient quality. So eventually we picked this 7th extraction method. And then the next step of this was optimizing the PCR. So PCR is this molecular biology approach to making many millions or even billions of copies of your gene fragment. And you need to do this in order to sequence. We need many copies of single genes in order to be able to sequence them. So this also required some optimization. We were using a protocol from the literature, but we had to optimize a little bit the annealing temperature for these primers and the concentrations of different reagents. But eventually, Matt and Joe were able to get nice amplification. So here's one of the gels where we see these nice bright bands right at that 4200 base pair length that we were looking for. So then we sequenced it on the nanopore. And then after that, you get raw reads. So it just looks like a bunch of ATCG. And then we have to process those as well into something meaningful. So this is where I developed a bioinformatic pipeline using some tools that are relatively new because they reflect the new technology that's been coming out, like nanopore. So the first step was quality control of the reads using these two tools called NanoFilter and NanoPlot. I'll also say that all of this software is open source software available for free from bioinformatics developers online. And then we got really lucky. At the same time that we were developing this project, this new tool came out called Mirror, Microbial Identification Using rRNA Operon Region. And so this is a tool that will annotate your long read sequences. So there's tools for this for the short reads that are really well established, but in this approach, you're comparing your reads to a database. And so you need a really clean database of the long reads in order to compare to. And that's what Mirror provides. So Mirror has a database of over 97,000 of these sequences that cover almost 10,000 species. And so we use the Mirror tool in order to annotate the species that we have in our samples. I will say 10,000 species sounds like a lot. In the microbial world, it's not that many, but this is just the first generation of Mirror. So in theory, the next time that they update this tool, there should be many more species. They're working on curating the database. And I'll also say that Matt and Joe checked the database for known oyster pathogens that we found in the literature, and they were there. The majority of the pathogens that we could find in the literature are in the database. So if they're in our oysters, we should be able to detect them. And then I used this tool Corona from, it's an open source NIH tool for looking at the breakdown of the different species in the samples, and also did some ecological analysis with a tool called Phyloseq and Vegan in R. So all of this sounds kind of complicated, but can you see that window that I
Yes.
just, okay, great. So all of this, the bioinformatics part and the DNA extraction part are all going to be open access online. So Matt and Joe have made videos of themselves extracting DNA and doing the PCR. Those are currently being uploaded to YouTube. And the bioinformatics is laid out in these online notebooks, so you could actually follow along and run it yourself. Okay, so let's look at some breeds from our oysters. So this was one of the first plots I generated showing the quality of the reads, which I got really excited about. So here is, on this axis is the average read quality, and on this axis is the average read length. And the first thing that popped out to me is that the highest number of reads were right at that 4,200 base pair cutoff that we were expecting. So I was really happy about that. I use those tools from the previous slide to filter these. So we filtered out the low quality reads, and we filtered out based on size also, so just plus or minus 20% of the expected length. So then we annotated what was remaining after that quality filtering. Then I did something called rarefaction. So the theory behind this is that every time you sequence another read, you annotate another species. You get another read, you get another species. You get another read, you get another species, until eventually you keep sequencing and you're running out of new species to detect. So in rarefaction curves, we usually see this, this threshold reached as you acquire more reads. You usually reach a threshold of a maximum number of species. And obviously some samples have more species than others, but the idea here is, are we reaching or approaching that threshold? And it seems like yes, using the MinION approach, we are at least getting towards that plateau, which means that we're sequencing almost the entire diversity of these samples. So it's not like we're missing a whole lot. So I was also very excited to see this. And then here's some abundance by different phyla. So here's some samples that we sequenced on this axis, and then their individual read abundance broken down by different phyla of bacteria. And I was excited to see this as well, because number one, this is the similar total read abundance that I would expect for second generation sequencers, which have those really high throughput. They generate, they can generate millions per sequencing lane, which when you divide that across samples, you get hundreds of thousands per sample. So this is usually what I would see when I use an approach like Illumina, and we're seeing it also with the MinION, which is great. We're seeing the same general amount of reads. This also made a lot of sense to us because B11, which had the least amount the least amount of reads was our negative control. So there's some contamination in there, and these ended up being all human associated microbes. So they're a contamination from us processing the samples. And then this one was our positive control. So lots of reads, but all within one phylum. So we put in DNA from one known species. And so that all came back annotated as one thing. So that's great. So the rest are our different samples. And you start to see just from the breakdown of the phyla here that some might be more diverse than others, right? So there's a lot more phyla in this sample than there are in these samples. So I calculated something called a diversity metrics, particularly one called Shannon's diversity metric. And so this is this sort of like collapses the whole diversity in one sample into one number, and that number represents how diverse or not diverse the sample is. So a higher number means more diverse, a lower number means less diverse, and this takes into account the total number of species, but also how well they're distributed. So you can imagine a situation of like, there's two samples, each of them has 3 species, each of them has 30 total individuals. But one sample has 10, 10, and 10, and the other sample has 28, 1, and 1. The first one is actually truly diverse. And the second one, it has the same number of species, but it's not as diverse because that distribution is largely swayed by one species. So this metric takes into account both of those things, the number of species and how they're distributed. And you can see that same sample that seems like it had high diversity here, B2, does actually have a higher diversity metric. There's many species there. So we can also break these down in something called Corona plots. So I'm going to focus in on these two samples for comparison because they were sort of these two extreme events. So this is sort of like a pie chart of the breakdown of the species in sample B2. So I showed you the phyla, which is this taxonomic level, but it goes further into family, order, class, and so on, all the way down to species on the outside of the circle. So you can see there's lots and lots of species here. Great. If we look at B3, immediately you see there's a lot fewer species, right? It's much less diversity, and the majority of the biomass here is only 2 or 3 species, which is really very rare when you look at environmental microbiomes. So what did we actually see in those samples? So that low diversity sample, this one that only had mostly these 2 or 3 species. These are the 2 major species that made up that abundance. So 34% of that low diversity microbiome, oyster microbiome, was this species called Lactococcus pyotori. This is a known fish pathogen. And what's really interesting about this, so this causes a disease in fish. There's been recent outbreaks in California, and specifically in California hatcheries, fish hatcheries. They detected this for the first time in 2020, and they're really concerned about it. They've actually had to put some hatcheries on isolation so that it wouldn't spread. And as far as we can tell, nobody's actually reported these in oysters before. So that was this big yellow one here. And then the second most abundant species in this seemingly infected oyster was this Photobacterium damselae, which was 28% of the microbiome. And this is also a known pathogen of marine animals. It's been detected in fish, crustaceans, mollusks, and cetaceans. So mollusks, including oysters. But those weren't the only 2 pathogens detected in this oyster. So there's a whole list here of known either opportunistic or acute pathogens of oysters that we found in this oyster using this method. And in that very diverse one, we saw a lot of beneficial microbes, which was really cool. So this one had a high diversity of lactobacillus species. So bacteria in the lactobacillus genus are actually probiotics. So if you ever buy probiotics, like the little pill of probiotics, a lot of them are lactic acid forming bacteria called lactobacillus. And they've been at least hypothesized to be beneficial for oysters as well. In particular, we found this known oyster probiotic called Enterococcus faecium, which is protective against pathogenic species. It's been shown to be protective. And then other species that are important potentially in biogeochemical cycling, which can happen around the oyster, specifically a lot of cyanobacteria that are important for nitrogen fixation, and a lot of sulfur cycling microorganisms that are involved in like sulfate reduction or sulfur oxidation. So this diverse microbiome really seemed to have not just diversity, but also lots of good bacteria. So just to summarize, we were able to build an efficient DNA extraction method, PCR amplification method, and the bioinformatic pipeline for detection of the members of these oyster microbiomes at the species level. All of these steps are documented in detail and reproducible, so they'll all be online. We can detect both beneficial species and pathogens, and there does seem to be a relationship between the diversity of a microbiome and its health. So like I showed you, that high diversity individual had a lot of the beneficial microorganisms, and a low diversity individual was dominated by the ones that are pathogenic. So in the future, obviously, we want to continue to screen more samples. And something that I'm really interested in doing in expanding a project like this is to start to establish thresholds. Like, we think we had one healthy oyster and one sick oyster, but the rest of our oysters were somewhere in between. They had pathogens and they had beneficial organisms. So at what point does an oyster actually become infected on the individual level? And beyond that, when does that become important for the population of oysters where they're living? So if one has an acute infection of multi-organisms that are bad for it, what are the parameters in which that becomes important for the health of the entire oyster reef, the population that's living near it? So that's all I have to say. I just want to quickly acknowledge a few people that really helped with this project. So I already mentioned Heidi and Lee from Rutgers. So both intellectually, they contributed to this intellectually and me thinking about all of this, but also Heidi helped us a lot with sampling. We coordinated a lot with the Billion Oyster Project, especially Jen and Tatiana at the Billion Oyster Project. The Molloy Faculty Scholarship and Academic Advancement Committee helped to support some of this work, but of course, most of it was supported by the Water Resources Institute. So I'd like to acknowledge you as well. And that's it. I will stop sharing.
Thank you, Liz.
Yeah, of course. Thanks for listening.
If anybody has questions, feel free to just unmute yourself and ask. We usually reserve this time for an informal discussion, or feel free to leave it in the chat and I can read it out for you.
Yeah, you can, if you have a question, you can say.
Yeah, just a question. Could you remind me why the 4200 base pair is important again?
Sure. So it gives us the resolution in the sequence in order to tell the difference between species. When you only look at a short piece, you can maybe tell the difference between different genera, different members of different genus, but it's not enough to tell the difference between species. So we need that entire length in order to differentiate between species, and even within the same species, we can differentiate between strains. And that's important for pathogens because, for example, the genus Vibrio has many beneficial microorganisms and many pathogenic microorganisms. So using just the short reads, we can't tell the difference between those, but using this approach, we can start to determine which ones are there.
Thank you.
Of course.
I have a quick question. Hear me?
Yep.
Maybe not. Oh, awesome.
Can you guys also
so-
identify who you are with your question, please?
Yeah. So my name is Josh. I'm with Cornell Cooperative Extension. I want to run a large scale production hatchery out in Southold. Do you have any interest in trying to sequence larvae, oyster larvae at all?
Yes. I tried to get some from I think somebody, Brian, you sent me up with, they were
Hair.
collecting, yeah, they were collecting wild ones in the Hudson, but they weren't able to find any this past year. But yeah, if you have samples, I'll sequence them.
Yeah, I do multiple spawns, so if you guys have interest, to give you guys 1,000, 10,000 larval animals is a drop in the water for us. If you want, I can reach out to you via email after this is over and talk to
Absolutely.
you about that further. Okay, cool, awesome. Thank you.
Thanks. Yeah, it's a really great thing to know because there's some work showing that infections also depend on life stage.
Yeah.
So here we're obviously looking at adults, but probably we'd find something completely different in the larvae.
Yeah, and it's interesting too, because if we have any unexplained die-offs and all the oysters that we grow and everything at this hatchery is all for restoration purposes. So at the end goal, we're planting spat on shelf for different sites, but it would help piece together the puzzle, especially this is something that I can do in-house without having to really, send it out to a lab to do the work. It's really beneficial. So thank you to all, and thank you for doing this work. So awesome.
Yeah.
Thank you.
Yeah, thanks.
Elizabeth, can I ask a question?
Sure.
So I'm Lewis McCaffrey from New York State Department of Environmental Conservation, Finger Lakes Watershed Hub. Just a comment, I don't think I've ever had such a literally visceral reaction to a talk as I've just had with yours. My stomach was flipping over hearing you talk about all of these bugs inside oysters, and I think it's put me off eating shellfish for the rest of my life. But more seriously, I wanted to ask, I was really interested about your description of the 3G technology. I wondered how expensive each sample is for a start. Number two, are there libraries of these genetic markers for other kinds of organisms, for instance, picocyanobacteria? And number three, it seemed like it was like a little device that was on your desktop. Is that where the human contamination came in, or was I getting the process wrong?
So, okay. Question number one was
It was how much, how expensive. Number two,
Okay.
other organisms, and number three, issues with contamination.
So, to get the system up and running, you have to buy the sequencer and an appropriate laptop, and that's less than $10,000. Once you have that, the only cost is reagents. And so I would say you buy the reagents for like 6 reactions, and that's probably about a few $100s, and then the chip to run the sequencing is $700. So if you want to keep, we're talking about, it's not nothing. It's a few $1,000s each run. And then each run, depending on which kit you bought, can give you anywhere from 12 to 96 samples.
Okay.
But as you increase in number,
right.
but as you get more samples in one chip, you get less resolution. So I use one that has that pooled 12 samples. I didn't want to go too high because then I would lose some of the diversity in each sample. So I think that the 12 is a good one, is a good place for this kind of project.
So it's about $100 to $300 per sample.
I would say so. Yeah, once you have the
Okay.
system set up and running.
Thank you.
Yeah. In terms of other organisms, yes, anything that is bacteria should be caught by this, including cyanobacteria.
Okay.
So the factor that determines that is the genetic tags that you use to stick to that gene. And so we're using bacterial primers. So anything that comes under bacteria should theoretically be here. And we did see lots of cyanobacteria in the healthy one. And then the last question, sorry.
Contamination.
Oh, yes. Oh, that probably happened in various points. So the actual sequencing, which does happen on that little thing on the desktop, I don't think it happened at that step, because by the time you're putting the DNA there, it already has like specific little things that help it stick to the chip and whatever. So if I was breathing on it, my DNA would not have stuck to the chip. But before that, there's a lot of like pipetting and moving fluids around that we try to do in a clean hood. Actually, I think I had a picture of Joe working in the clean hood. Yeah, really quickly, I can show you this again. Oh. So this thing here should control most of that contamination, but it's impossible, you know. I mean, first of all, you have to collect the oyster in the wild, right? So somebody is picking it up out of the water and putting it in a bag, and as clean as you can be, somebody's breathing on it. But I mean, I've done a lot of microbial ecology work for other things, and there's always some level of contamination. You just basically have to make sure that you run blanks and then subtract those species out when you do the analysis.
Thank you.
Yeah, you're welcome.
Sandy asked in the chat if this will be recorded and if we'll be able to view it later on, and it will. We are recording it and we'll probably post it on our public YouTube channel. So if you registered for this talk, you should receive a link to that soon.
Liz, I wonder if you could comment, you know, earlier I set it up from the, so I know it was like from a maybe a very high level, these sort of overall ecosystem and estuary management plans that talk about wanting to assess restoration efforts. How important, or, you know, what would you say about, you know, pathogens and the idea of understanding and maybe controlling pathogens? How would you say that like fits in the context of other stressors or things that might be hurting restoration efforts in general?
Mm-hmm. So one of the major hypotheses that we had going into this was that temperature was a really important control on the abundance of pathogens. And, you know, we don't really have enough samples to say whether that's true or not yet, but I would like to start to answer that question with more sampling. So the idea is pathogens in general, not just bacterial pathogens, but like if you think about like ticks and mosquitoes, they are prolific as temperatures warm, right? And so we have a Hudson River right now that is warmer than it used to be. So potentially there are just sites that are no longer maybe suitable because the stress from pathogens is too high. So a tool like this would help to determine which of those places maybe that pathogen stress is just too persistent versus a place where oyster restoration might be more successful because maybe water temperatures are a bit cooler or whatever other parameter there is controlling pathogens. Does that answer your question?
Yeah, I think that's good. I think, you know, like understanding the world of pathogens and their detection and quantification can be challenging, right? But I think making a link between that and the idea that, like, well, warmer water leads to more bugs, right? More pathogens, or can. And that's something that this is potentially useful for. So thanks.
Yeah. And what's really great about an organization like Billion Oyster Project is they use adaptive management, right? So they go out, they try something. If it doesn't work, they modify it for the next year. They go in a different place and they try it. They try the same, or they try something slightly different. So they set themselves up to be really modular, and so they can respond to information as they get it. So if they had a tool like this, they could potentially, you know, pick those sites that are more appropriate.
Maybe one other, you know, somewhat like definitely tangential question. You know, like the Billion Oyster Project has a lot of publicity around it, right? And so there's there are other, you know, approaches to, you know, to preservation and sort of causes that don't necessarily have the highlights that the Oyster Project does. do you have anything, like, how would you say that organization, like, has it helped to shed light on the science? Do you feel like, I guess I'm just a general question of, like, when you have such a strong and, you know, a strong advocate in the news, how does that change? Does it change the way you do science? Does it give you opportunities you didn't have? Does it cause a headache sometimes, right? Just wondering if you ran into anything like that.
Not necessarily. I mean, I don't even remember how I heard about them in the first place, but what's been really great is that they employ scientists, right? So Jen, who I acknowledged in my talk, is an ecologist, a marine ecologist. And so and I've worked with other scientists through them as well. So they are very interested in not just in they do a great job with the outreach, and a lot of their reefs are community run, so they're also really good at getting community members involved in restoration, and they do a lot of educational, especially K through 12 educational activities. But they have people dedicated to doing this according to the science as well, and so they're very interested in pairing with scientists from academia and so on. So it's been really great working with them. Yeah, and they're very willing to, like, you know, if I talk about, oh, I was hoping to have temperature data from this site, and they're like, oh, maybe we can put that in our plan for next year. You know, like they're very interactive, and they want to help scientists.
Thank you.
I see a question in the chat. Sandy asks, are there other schools doing this sort of research in different areas to be able to get a large pool of data?
There are certainly a lot of people interested in microbiomes, and there are a lot of people interested in oysters. On the West Coast, they have a different oyster species. It's not the Eastern oyster, it's a Pacific oyster. They do a lot of oyster microbiome research, I think, through the University of Washington. Here, you know, there's a lot of interest at Rutgers, the scientists that I mentioned that I acknowledged earlier, we've already sort of joined forces, and we're kind of trying to work together. There's also interest in developing probiotics for oysters. So like at UConn, the NOAA lab, I know they're interested in looking at the microbiome and trying to pick out the good guys and grow them in order to help hatcheries and things like that. But yeah, I don't know. There's probably other groups, but I'm not aware of them.
Does anyone have any last questions? Any questions in the room?
I guess, I just have a very general question about the process of, restoring an oyster reef, like what those steps look like and how long it takes.
So it's really variable, actually. So they have all of these different, I don't do this. I just can speak to what I've seen the groups do. So they have a lot of different types of structures. So they'll have like I forget, they have all these names for them, but they're kind of like cages, right, with like different components, and then they have different ways of installing them. So some will be like sitting on the bottom, some will be on a buoy. They'll also try like different cement structures that have a particular coating on them that oyster larvae like to settle on. So they try a lot of different things, and I believe that Billion Oyster Project finds that different things work in different places. The different structures work in different places. So it is all really like, it is a restoration practice that you really have to be adaptive at like the micro scale in order to get it up and running. But they
Yeah.
essentially, they have a hatchery where they grow them. And then they'll usually like have them settle on shell and then put those shells in the cage and whatever the cage is and put those out. And then they go back year to year and they count how successful those guys are.
Thank you for explaining. And then they just leave those cages out there, or they remove the cages in time?
They usually, no, they leave them. So the idea is that they want them to, establish into a wild oyster population. So eventually that those oysters will now reproduce and sort of spread, in that lagoon or wherever they are. And so they also look for natural recruitment around the restored the structures. Yeah, but the idea is to leave them. They do remove them sometimes if something is going wrong, but the idea is to leave them there.
Okay, I don't think I see any more questions in the chat, and I'm assuming there's no more questions from the audience. That was a great talk, Liz. Thank you so much for
Yeah. Thanks for
presenting.
having me. It was a lot of fun.
Thank you.
Thank you, Liz.
Thank you.
I will still be eating oysters.
Me too, actually.
Thanks, everyone. See you next week.
Bye.